This symposium is a pivotal moment on our collective journey towards a more quantitative approach in genotoxicity assessment. Genotoxic compounds are compounds that can affect DNA. Either they can do this directly or they can do it indirectly. There's a very old paradigm, and this paradigm says that one single molecule might be able to induce detrimental effects to human health. This has led to the situation that we can at the moment not identify for humans a safe level of exposure for these compounds.
Traditionally, we say genotoxicity is something where you can't work with quantitative terms, right? So you can't say this is a little bit more or a little bit less genotoxic. If you work on a pharmaceutical molecule, on a new drug candidate, for example, you say, okay, listen, this is genotoxic, so I can't use that anymore, I throw that away. Okay, and I don't develop that. Well, that works for, as I just said, for the pharma industry, that may work, but for other, say, situations, it may not work. For food ingredients, for example, or for environmental situations, for molecules in the environment, you can't avoid very often an exposure.
The ambitious goal of quantitative genotoxicity assessment is to extrapolate genotoxic markers that you would have from cellular or molecular models to the dose-response relationship that should reflect the whole organism. So I think this is quite an ambitious goal. We are assisting to a decreasing number of chronic animal studies, and this is a great problem. We need an alternative approach really to do risk assessment, and quantitative genotoxicity is a possible strategy. We have been listening this morning to fantastic development of new experimental assays to detect mutation in vivo. So we start to have the tools that I think really will allow to do a much better quantitative genotoxicity assessment.
I'm convinced that a quantitative approach in genotoxicity assessment is urgently needed. However, the task of working out of how and if this is possible is daunting, as are the regulatory challenges. That is why I believe it's important to start a discussion now and to do so in an open and transparent manner.
Well, the greatest challenge is, of course, you can define a substance as being genotoxic, but the point is, what is the real risk for human health? And to quantify this risk, that is of a challenge, because there's so much difference between these genotoxic compounds that indeed a better quantification of the risk would be certainly helpful for the risk managers.
The quantitative risk assessment of genotoxic substances still deals with major scientific uncertainties, and also these uncertainties pose a question on what is the level of risk that society can accept.
The quantitative approach adds a tool. Whether it's needed is a question of when it's useful. We need to explore how useful is it in the identification and prioritization of risks, and where can we agree, because at the moment there's no internationally accepted and agreed guidelines, and that certainly would be the way forward to discuss and agree what guidelines we can bring forward for quantitative risk assessments, because it definitely has a place.
I feel what we learned here at the conference is that especially the colleagues from industry are quite advanced with respect to the toolbox. Even so, risk assessment should always be independent. This still allows us to take the scientific ideas of industry on board, and I feel we really should do so.
Frans Verstraete, when you are demanding a scientific consensus, and you just mentioned that you talked to your colleagues in Brussels yesterday, is there a consensus among you?
From the moment there is indeed no consensus in the scientific world about a certain approach, then indeed there might also be the reluctance from the regulators to change.
I feel we cannot wait. Towards the quantitative approaches, yeah, why not give it a try for the contaminants? People might accept the quantitative approach for the contaminants because they are not avoidable. They are out there, but they might not be ready to accept this for regulated products like pesticides or food additives. And maybe it's good to give it a try with the contaminants and let people know, look, it's better than to do nothing. And then we can decide at a later stage, maybe in 5 to 10 years, how to do this with the regulated areas.